So now we are at the home page of ENSEMBL EBI. We are interested in the services menu. So the services page has mainly two resources, tools and data resources. So the tools are Cluster Omega, Interpro Scan, BLAST, etc. We will not be using any tools from ENSEMBL EBI during our lectures. However, we will be using data resource, especially ENSEMBL. So let's go to ENSEMBL. Now we are at the home page of ENSEMBL database. We are interested in downloading protein sequence from the ENSEMBL database. We will paste the name of the gene, and as soon as we do that in the search bar, we see that several links drop down. However, we'll just go and click on the search icon. Number of options appear. We are interested in the human gene, so we'll go and click on the human gene. Now you'll see two tabs here. One refers to the location of the gene, the other is the one that we opened, and that is, it gives the abbreviation of the gene, which is ADCYAP1, followed by the ENSEMBL ID for gene. Then there's a description, that is the full name of the gene, another synonym for the gene, and several other informations. Importantly, we also see that there are 4 transcripts and they are shown here in this table. This is the name for each transcript. Each transcript has its own ID, and then we get the information about the number of base pairs, the length of protein sequence, etc. What we want to do is we want to pick the topmost transcript with the highest number of protein residues, amino acid residues. So we'll go and pick this one. Now before I click on this one, let's have a look on the left side here. This is a gene-based display. So you have several options and sub-options. So there's a summary that we already see, the splice variants and transcript comparison. Then in sequence, you have secondary structure. There's comparative genomics that talks about several other aspects like genomic alignment, gene tree, gene gain and loss, orthologs and paralogs. This is an important information for us. We will come back to it later. Then you see gene ontology information. So what is the cellular component where this gene is getting expressed? What biological processes this gene might be involved in, and what is the molecular function of this gene? Now let's go and click at this transcript. Have a look. This tab is still there, and a new tab has opened. And this tab now has the number of transcript that we picked. And the left menu has changed now. So now this is transcript-based display. What we are interested in is the protein sequence. So we'll go and click there. You see the protein sequence. We want to download the sequence, so we'll go and click download. So this is the file name with which the protein will be downloaded. This is a file format. We want it to be in FASTA format. We are going to go and let's make sure that it's checked amino acid sequences. It is amino acid sequences. Download. It will ask you where you want it to download. Let's download onto desktop. Save. It has been downloaded. Now, what we want to do is explore the orthologs of this particular gene. So let's go and click the Gene tab. Let's go to the orthologs. And you see within the Gene tabs, now there's this ortholog sub-window that's opening. It's taking time to load. This can happen with ENSEMBL. Sometimes it takes time to load. Now it shows there are 133 one-to-one orthologs in 200 species. we are interested in one-to-one orthologs. However, we are only interested in other primate species. So I'm going to go and click this. Now, if you move down, you will see name of a primate species, type, it is one-to-one. Then the ortholog name, which you expect it to be the same as the gene name that you picked. It can also show you already the sequence alignment, but we want to go and check that for chimpanzee. That's the closest relative of humans. You get this option to check view protein alignment, view cDNA alignment. We are interested in protein alignment. Let's go and pick it. Okay, now you see one-to-one alignment. This was done using cluster W tool that you saw earlier. Now, let's try to read this alignment. The top one, ENSP, if you look, that's the peptide ID for the human gene. Then there's a backslash, and then the number of residues, so from 1 to 176 for the human gene. Then the next one, the bottom one, that's the peptide ID for the chimpanzee gene. See here, same backslash, number of residues for the chimpanzee gene. And then you see the amino acid residues of these. So the star represents identity. So every time you see star below these, you see they're identical. Then there are dots that basically represents some level of similarity between these residues. When there is a gap, there's nothing else. That means that they are neither identical nor similar residues. We just wanted to check how aligned they look, and they look pretty well aligned from the first to last residue. There are some substitution that we observed, but largely they are identical. And when you check what is identity, it's 90%. Let's go back and check this identity with other species. We'll again go and click on primates. This time I want to select gorilla. Again, you see percentage identity is 90% for both sequences. So the identity between the human and chimpanzee protein and human and the gorilla protein similar. Let's go back once more. Now I want to pick the gibbon. And here you can see that the identity is 1% less between human and the gibbon protein. Now we can download homology here. Basically, it will give us the pairwise alignment, but we don't want to do that right now. What we want to do is download the FASTA sequence for orthologs from all three species. So what we'll do for that is go back. Check unaligned protein, check FASTA format, and download.